this is now a close-up of cavitation and boiling occurring in a phantom tissue. This is a phone, a fiber optic hydrophone, and the cavitation is going to occur in this gel in this vicinity. And so here's the movie, and you can see in a few milliseconds you get this big bubble here. That is boiling, and it's hard maybe to see here, but this is cavitation. So let's look at it in a little better frame. So now here is the hydrophone, 800 microns, just about a millimeter in diameter. This is the focus of the acoustic field where you're generating the temperature growth, but prefocal, because the negative pressure actually occurs at prefocal. All these little spots here are individual cavitations. So in this particular sense, we have started out, and very soon we get cavitation, and then after a certain period of time we get boiling. Now, to analyze it in more detail, we look at here. So this is 3.8 milliseconds looking at the focus, and this distortion here means that you've distorted, the temperature is rising, and so you actually distort the field somewhat, but there's no cavitation yet. And then at 3.85, 5/100ths of a millisecond longer, you get individual cavitation events, and then at 3.9, another 5/100ths of a millisecond, you get boiling. So you see that you're heating the tissue, you get cavitation, and then the cavitation initiates boiling, and so one can monitor this with high-speed cameras and so forth, and analyze the difference between cavitation and boiling, because they have different mechanical effects. So the PCD here, as you can see, shows cavitation and then a very abrupt change, and that's boiling. Boiling is probably not desirable in many cases, and so one wants to notice that you want to avoid that. So one of the applications of high-intensity focused ultrasound is the Sonoblate 500 from these original company Focus Surgery, and these are used to treat the prostate for both cancer and for BPH. So here is a system. This is a transrectal system. The transrectal system can be pushed against the prostate, and this is an image of the prostate. And then you apply high-intensity focused ultrasound through transducers in the probe itself, and you can pick areas of the prostate that you want to ablate thermally, and this outlined in this red area here, but you can see two white spots. These are what we call popcorn, because what happened there is you got superheated boiling. That is to say, you want to heat the tissue up so that it denatures the proteins and destroys the tissue, but if you heat it up too fast, you get boiling. This is sort of like a superheated boiling. You kind of explode, and then there's all kinds of, you can hear this in a human patient, this sort of explosive boiling in the prostate. And these cavitation and boiling generate, cavitation-induced boiling can be problematic in terms of treating the tissue. So there is, when you apply acoustic field to tissue, you have two things going on, or could have. One is you're going to heat the tissue up, as we have demonstrated here. And you can model that quite well. The thermocouple measures the growth of the temperature, and you can have a theoretical model that you can model it, and you see that it treats it very well. And then you turn the field off, and the temperature goes back down. So this is a way that you heat tissue. Now, if it turns out that no cavitation, no boiling, no hyperecho, but if you suddenly get cavitation, which you can measure from the PCD, now not only does the temperature change, it could go much higher because the cavitation could be scatters the sound off the field, get nonlinear effects, and you actually can have a very sharp rise in temperature. But also, in this particular case, the temperature would go down because you scatter the sound field out and you're not getting the extra heating. This is atypical. Normally, the temperature would go much higher around this particular point, but it shows the interaction and the detection of the point of cavitation when you're doing heating. So this is a little bit getting into some sophistication in terms of monitoring. Now, how else can cavitation have an effect on tissues? Well, most of you know that there are ultrasound contrast agents. They are stabilized microbubbles, and when you put the stabilized microbubbles into tissue, then they give you contrast, and if you increase the pressure, sharp pulse, you can destroy these contrast agents, and then you can see their regrowth as they perfuse into tissue, and then you can use this in cardiology to detect areas of infarctions and so forth. Anyway, these bubbles, contrast agent bubbles, can be used to introduce or activate a biological effect in tissue, and then you can monitor this with diagnostic ultrasound. So let me give you a couple examples of that. One of them is called sonoporation, in which you want the acoustic field to transmit a DNA segment or some drug into a cell, and then the cell, we would like in some cases it to still be alive, and then it could produce that effect or represent that by certain effects, like it could produce a particular protein that would be used, for example, to treat some disease or something. So here, these are cells that have not been acted on by acoustic field. Here are some cells that have been acting on by a field, and they're actually producing a particular protein that shows in the color here. Now, so this is basically what physicists like to talk about, its mechanism. So this video from Alfred Yu is, this is a cancer cell. It's plated on the surface of a glass slide or something like that. And a single bubble is introduced right here. And we're going to show in this movie, as the bubble is hit by a short pulse of ultrasound, how it will cavitate, break apart, and then it'll induce a hole in the membrane. So let me show you the movie first. So there is the bubble, and you see it popped. That is to say, there was essentially one small pulse. The bubble doing its biological or doing its mechanical thing introduced a hole in the surface. Let me back that up and show that again. No, it's difficult to see that. So here's another couple of cases where these bubbles are used to introduce drugs inside tissue. You can have drugs that go into tissue and destroy the tissue, or you can have DNA fragments, plasmids and so forth, to go into cells for them to generate in the living cell a particular protein. So you see, in this particular case, propylium iodide is the drug, and the bubbles on the surface. And let me back that up. This is a very nice movie to show that when the bubbles are these little green things here, and when they're activated, as they oscillate, they tear holes in the membrane, and then the propylium iodide goes inside the cell. And so this is site-specific drug delivery, and using contrast agents as kind of a transformer that transforms the energy of the acoustic field into a microscopic hole in the cell itself. So one of these things that are now fairly active, moving toward clinical effects, is that, as you know, blood-brain barrier protects our brain from the infection from various pathogens and big molecules and so forth. So the tight junctions along the blood-brain barrier also protect our body, but they prevent us from using a drug to go in and then treat, let's say, some kind of cancer inside the brain itself. Now, if you introduce bubbles into the vessel, into the blood, into the medium, and then you use ultrasound, you can actually cause these tight junctions to be opened up. And Hong Chen and her colleagues at Columbia University have been able to demonstrate that by using a focused ultrasound and ultrasound contrast agent, you can introduce various relatively large molecules inside outside the blood vessels into the parenchyma of the tissue itself. And these data show that at different acoustic pressures, you've got different inputs or translations of these drugs into the brain. And in some cases, if you want to get these 2,000 kilodaltons you see here at this particular pressure, 0.84, it's very difficult to get these big drugs into the brain. And you need to get, in this particular case, it's kind of complicated, but you need to get inertial cavitation. SC means stable cavitation. IC means inertial cavitation. So if you apply pressure at different amplitudes, then you can get these drugs into the brain, and you get the mechanism, the physical mechanism that permits this is this cavitation. So cavitation can be a useful mechanism in order to treat cases of passing through the blood-brain barrier. What's the mechanism, again, for opening up these holes? Here is a movie that Professor Kudo gave me, and this is a cell, a relatively large, I think it might be a cancer cell of some sort, and there is a bubble there, and a bubble there, and here, right here. And when a pulse hits that, it will knock a hole in the bubble, so in the cell. So look at these little bubbles here, and the arrows are going to show. Now here comes a pulse. It might be difficult to see, but there are holes. There's a hole there and another. Let's see it again, and maybe it'll be better. Bang. Well, it depends on how visible this is to you. So what's happening there is that the bubble could be producing a liquid jet, and the liquid jet could be knocking little holes in the cell wall. And Tachibana published a paper when he had cells, and you see all these little holes popped into the cell there. And this is some indication that these jets are causing damage. On the other hand, if you had acoustic streaming, if you had stable cavitation, this is, for example, inertial cavitation. If you had stable cavitation, then the stable cavitation could also, in the vicinity of the membrane wall, could tear the membrane. And so either inertial cavitation, jetting, or microstreaming could be opening up, and people are not so sure, and probably both of these things are involved. Now, a couple of final things is that you can use cavitation and high acoustic pressures to ablate tissue itself. So that's called histotripsy, the breaking of the tissue by the cavitation itself. And so in this particular case, use maybe 5 or 6 cycles of enormous acoustic pressures. This is on the order, maybe, it's very hard to measure something like this, but maybe 180 megapascals. That's 1,800 atmospheres of pressure, but only for a few cycles. But when you do this, and this is some tissue, and now they focus the histotripsy focus, transducer focus this particular point, and you can see now the cavitation occurring at a particular point, and it's eroding the surface of this tissue. This is perhaps better seen in this movie, where there is a membrane here held. This is a heart membrane, and now they're focusing on a spot right in here. And as you see, as the time goes on, a change in the surface of the membrane, and after a few seconds, I don't know how long this took, you actually cut a hole in the membrane. And there are certain cases where the membranes in the heart need to separate the chambers of the heart, need to have a hole in them, you know, the clinical application. So not only can you destroy tissue by intense cavitation, which we call cavitation histotripsy, but some colleagues in my laboratory at the University of Washington, Vera Koklova and her colleagues, have shown that boiling can also introduce this these destruction of tissue. And so in this particular case, showing you this is some ex vivo tissue and a focused transducer, and here are the numbers and so forth. And what happens is that now we actually generate holes in the tissue from boiling. And let me go a little bit further to show you what this is. So if you have these particular pressure amplitudes, and these are no longer 180, you can reduce it down to the moderate of pressure amplitudes of 50 and 60 and 70, and you get now holes at different sizes depending upon the various amplitudes and so forth. Now, what is happening here? This is kind of complicated, but let's suppose that you have an interface, and this is liver tissue, and you focus the HIFU source, and these are the parameters, the HIFU source, right on the interface. What happens is that you get jetting. You actually, the tissue is destroyed and it's blown out into a shower. So this is the emulsification of tissue by a focused ultrasound on the surface of an interface. Now, how can you produce then this hole in tissue? Well, first of all, if you have boiling, and this shows you a hole that's cut into liver tissue. And now, if the hole that is generated by a boiling bubble that lasts for several milliseconds, and now if you have a megahertz frequency ultrasound, you have those cycles coming in while the hole is expanded, and what you get is you get a blowing of the tissue into the hole. So the reason these holes are generated from boiling is that you're blowing the tissue off the surface. So finally, one of the most appealing things of therapy ultrasound, I think, is that this stimulated immune response. We talk about now the real frontiers of what's going on in medicine is stimulating the immune system to treat cancer. One of the things that people have investigated is, can you stimulate an anti-tumor response by stimulating the immune system? Now, one of the problems with stimulating the immune system to treat a particular cancer is that cancers have evolved so that they hide their characteristics, their molecular characteristics, from the immune system itself. And so if the tissue is not disrupted, where you expose the antigen to the immune system, then it cannot generate an immune response. But when you break up the tissue, as you would in histotripsy or boiling histotripsy, and expose the actual molecular species to the immune system, then you can stimulate a response. And Professor Bu in Chongqing treated a patient where he had lung metastases, and he then treated the original primary, which was a osteosarcoma, and then he saw a stimulated immune response, and these metastases in the lung went away. This is just some evidence that it can occur, and Professor Bu examined this on a molecular or a biological level, and here's the tissue that was destroyed by the HIFU, and then there is lots of lymphocytes that are directed to this, and they are causing the tumor, the live tumor in a sense, to be treated by the immune system. So that's a review of the fundamentals of cavitation, and that is show you some of the applications of cavitation in medicine. This is an exciting field. There are lots of frontiers to be generated and to be approached and solved. And I think that cavitation has a very important role in medicine and in the treatment of diseases. Thank you very much.